Well, good morning, everyone. Thank you for joining us for the EMSL Xchange Seminar Series. This is an opportunity for academic PIs to interface with EMSL experts about their research interests to explore how EMSL might help further their scientific endeavors. So I'm very pleased to introduce Dr. Jessica Gutknecht, who is an associate professor in the Department of Soil, Water, and Climate and a fellow of the UMN Institute on the Environment. She has a PhD in soil microbiology from the University of Wisconsin, and she is a soil ecologist with years of experience understanding how soil and soil biological communities mediate ecosystem responses to climate change in both natural and agricultural landscapes. And so her talk today will cover perennial grain systems as a pathway to improved soil functioning and climate resiliency. So thank you, Jessica, for joining us.
Thank you. Should I go ahead and get started?
Yeah, that'd be great.
Yeah, thanks for that kind introduction. And I was kind of saying this informally before, but I had a last minute shift to having to be home. And so the way that I'm screen sharing, I'm like really blind. And so I'm just kind of going to imagine all your faces and and I'll stop sharing as soon as my slides are done. And also, yeah, Sarah's going to help monitor the chat or the questions as we go. So yeah, and thank you so much for having me today. And I'm I guess in the same spirit, yeah, it's disappointing that I couldn't actually be there in person. So maybe someday. Yep. And so my title today is Perennial Grain Systems as a Pathway to Improve Soil Functioning and Climate Resiliency. I'm gonna also bring in some work that my lab is doing, not only on perennial systems as well. And you'll see that a lot of what we're doing is kind of comparing perennials to other systems and how we, then think about the soil resource and ecosystem functioning. So first, this is just a little screenshot of my lab and lab webpage. We also, soil and ecosystem ecology and climate change are really what's at the center of everything. And I have an ecology background. And so we also, there's some work going on not only in agricultural systems, but these days, what I'm going to talk about today is definitely, the main, one of the main focuses in my group. And so first, I want to just give an introduction to the bigger picture ideas, the environmental quality of food systems. This is probably something that everyone's quite familiar with, and just to kind of orient to some of the work going on in Minnesota. A lot of our rural municipalities are paying, over $1,000 per household per year to mitigate nitrate pollution, and especially in our sandy soils and some of the landscapes here, that's water quality and access to clean water is a huge, huge issue. So that's been one of the motivating factors of this work. Also, cropping systems are facing increasing risk of erosion, loss of soil and nutrients. Climate change is kind of upping the ante on all of those equations, including the last point, which is this world of food inequities, and producing enough food to get to those in need. And so kind of all of those together have been shifting a lot of people to think about the ideas of sustainable and ecological intensification. And so the idea is that, are we increasing the efficiency of inputs, minimizing nutrient loss, really improving the energy and input efficiency wherever we can at the plant scale, and then at the farm and landscape scale, maintaining ecosystem services and biodiversity, minimizing habitat loss. And then at the kind of global scale, it's of these, maximizing environmental values, accelerating rates of change, minimizing trade-offs when we're thinking of this global kind of food system and what that looks like. And not just, on the farm side, but this whole kind of gate to plate part of it when food leaves the field and getting to people in that piece of the ecological intensification too. That's a little bit less in my wheelhouse, but it's definitely work that I'm touching on more globally. And then, this then is often the translation of those principles where we still have these annual cropping systems, and we're just trying to improve the efficiency of them, reducing the, increasing the water holding capacity and the water throughput to mitigate flood and drought, reducing inputs, and improving the efficiency of those inputs. And then they have this little squiggle at the bottom of how we're improving the life in the soil. And so that, yeah, again, that's the way that people are typically, on the ground, like translating these principles of ecological intensification. But if we really, given the crises in the world, like, would it be a bigger lever if we really rethought food systems? And so what does it look like if we start to put perennial grains on a landscape? And this is a paper by Matt Ryan and some colleagues from a few years ago, talking about the kind of multiple benefits of perennial systems. And today I'm going to introduce you to a perennial grain that's kind of new, relatively new on the scene. And so it would still give the same, need for grain production, the need for an economic benefit to farmers who may not be able to afford putting their land, aside into buffers or into like CRP land. This gives an economic benefit, but it also, because it's a perennial, it might offer those benefits of perennial systems, deep roots. It's pulling up nitrate, nitrate and nutrients, and it's really, starting to repair and revitalize this system. So this is the way that, my group and my colleagues have been starting to think about these systems. And so today I'm going to give this in kind of 3 case studies that 3 graduate students in my lab are working on. And the first is not necessarily perennial agriculture, but, how can we better understand like these principles of soil health in the first place? Because the results are often really, really varied and hard to kind of navigate. And so, how are we understanding soil health and soil ecology with with kind of more regenerative farming in the first place? And then there are 2 ways, that 2 of the examples I'll show you about perennials. The first is the soil characteristics. When we look at these different cropping systems in the same kind of environmental conditions, and then start to layer on climate change. And then the third is the way that we're using isotopic tracers to then kind of mechanistically better understand the carbon flow in these systems. So those are the 3 examples I'm gonna show today. And I hope that there will be some time left over for questions also. So the first are these soil health surveys across farms in Minnesota. And this was a NRCS SIG grant funded in partnership with the Water Resources Center at the University of Minnesota. And part of the drive of the SIG grant was that was the study of these things in cold climates like Minnesota. So here, many people are very hesitant to reduce tillage or to grow cover crops or some of those basic kind of typical soil health building practices, because we have such a short growing season, and there are a lot of perceptions about tillage to warm the soil, or it's kind of a guessing game about if you'll have a cover crop that even does well here because of the short season for it to exist in the bigger season of typical annual crops. So that was a motivation of this, but what we've leveraged is this deep understanding of how these soil health practices change on the landscape. So this is the basic goals. We're collecting state soil health data statewide with a suite of standard methods. We are establishing a public soil health database together with the Minnesota Office of Soil Health, whose advisory board I sit on. We're analyzing soil health data from a regional perspective and also better understanding of variance with space and time, and then increasing knowledge sharing and networking around soil health. And so I have a slide at the end that kind of puts some numbers to the number of different partners we have in this work. And the partnerships throughout all of this are very, very key. I think, back to those big issues of ecological intensification, my belief is that, the time has gone for a scientist to sit over here, a farmer to sit over here. We really need everybody's head and spirit together to tackle some of these big issues. And so that's the aim of a lot of the projects that we work on too. So this is the regional differences in Minnesota, just to give you all a perspective of this. It varies quite a bit across the state. The normal, the soil suborders, which has been a big focus of this project, you know, how we can kind of get at the soil taxonomy with this work and line ourselves with that, with those characteristics. The normal annual precipitation, we go from, you know, we go from the northwest, which is, you know, close to kind of the Great Plains and more arid agriculture, down to very, very, you know, more wet precipitation. And then there's also this kind of opposite variance of our normal growing degree days. And then we also have, you know, not the beauty that you all have in Washington, but we do have topographical variance enough that we could really look at it. And this might actually be interesting for you, that how we looked at it and some of the results we found, even in relatively subtle changes in topography, have been a focus of this work as well. Our study design is that we found a set of farms in the same region, and we found fields really close together that are on the same soil type, same slopes, and we could compare conventional with soil health systems. We also always had a natural benchmark, and we measured, as I said, a wide range of physical, chemical, biological aspects of soil health. And so this is the one in the kind of orangish coat in the middle is Hava Blair, who's the PhD student working on this. The guy sitting on and, you know, that's me in my green hat, and the guy sitting on the ground is a local NRCS person. And we're kind of sitting on the fence row that would have been our undisturbed site in this case. And then we're on the conventional side, and then just over that fence is a certified organic farm that we did the soil health practices on, and the two happen to be cousins. And so we were able to find a lot of, you know, based on existing relationships, we were able to find these pairings across the state. So here's just a more specific, like what this looks like in these three categories: the conventional, the soil health, and the undisturbed benchmark. And then in each field, again, this might look relatively subtle, but with the soil forming principles that we tried to adhere to, you know, there's always kind of a summit, a shoulder, a back slope, a foot slope, and a toe slope. And so we actually ran at every single field, we ran transects at these different hill slope positions to ask, you know, in the end, these multiple layers of understanding the variance in soil health principles. And so if we're on the same soil type, you know, how does management impact things? And then also, you know, do we see variance, you know, between fields, between regions, and then within fields between these different landscape positions? And so one way that, you know, so now we're starting to parse out these specific management treatments. And so the data I'll show on this is regarding the change in tillage. And one of the ways we're looking at this, and this is the percent stable aggregates. One of the things that we found was changing more. And when we look at the average annual tillage events, what we see is indeed a relationship between tillage and the percent stable aggregates. And the SH, the orange, are soil health, and the CV is the conventional management. And so again, you can kind of squint and see that you have this difference. You know, we do have more big stable aggregates, which is what we want to see in a healthy soil, with less tillage. So that relationship, you know, we still see a lot of variance. That relationship is holding. But then what's really interesting is that we see that this relationship is changing across our different regions. So we have this Northwest, Central, Southwest, and Southeast regions. And you know, and so more or less that's holding, but the slope and so the degree to which tillage is influencing this is changing across the regions. And then what is really interesting, oh, sorry. Yeah, so it's the next data where this other kind of interesting thing is happening. So now we'll move from, you know, stable aggregates to one of our indicators about carbon cycling. This is POC. It's a very labile, kind of available active carbon source, or, carbon pool in the soil. And here we're kind of roughly seeing the same pattern with tillage, but, it's definitely a lot more variance and not as, and not as consistent of a regression. But in this case, what's super interesting is that the management was not as important as the hill slope. And this is what we're seeing across a lot of the data sets. So some of them, like aggregate stability, we're seeing these really consistent patterns. But in some cases, like with POC, we're seeing that, the upper or lower hill slope, especially in the Southeast, is really driving things more than management. And so here now you have this intersection when you're trying to understand soil health of like, what are you measuring, and how are you measuring it, and what are you comparing, and have you taken samples on the same landscape position or been thoughtful about that? And so this is how we're beginning to, in the graduate student work, Hava working on this has gotten some great like data analysis and data modeling grants where she's starting to tease apart all these different factors. Yeah, and here's another look at this where, we see a big change from hills, we see a big change from where we have our upper hill slope to our lower hill slope, and a much smaller change when we're going from this, conventional to the soil health management. Just to kind of emphasize that point one more time. Yeah, and so just to put some words to what I was saying, some metrics are responding as we'd expect, and then the context and variability are really important to our understanding. And this is even within one state. And so, and so the kind of getting at that nugget, I think, is going to be a good contribution to the literature where soil health is this, raging topic. And this, as I promised, is our number of participants. SWCD are the Soil Water Conservation Districts in Minnesota, the NRCS, the Board of Water and Soil Resources, 6 university departments. So this is really a huge effort across the state. All right, so now we'll move into more of the work on perennials. And so my kind of second chapter is the soil characteristics under different perennial and annual cropping systems in response to climate change. And the data I'm going to show you are from phase 1, because we're still actually at the end of the season for phase 2. And we even have one more round of soil sampling to take this year. So phase 1 was just to we did this in a different way. We rolled out these different cropping systems for 2 years. And then for the 3rd and 4th years, we're overlaying the climate treatments. And so the master's student working on this is just kind of comparing these systems and has helped us in this first year of rolling out the climate experiment treatments. So first I'll introduce the perennial grain that we're working on, Kernza. This is where if I could see any of you, I would ask for a show of hands, because actually even 2 days ago, there was another a PBS Marketplace did a spot on one of the Kernza farmers in Minnesota. So there's more and more kind of pot press about this perennial grain happening. And so more and more people have been hearing about it. It originated and was imported as a forage grass roughly 40 years ago. And the Rodale Institute, and then Jerry Glover, who's pictured here, and the Land Institute, and now the University of Minnesota have been kind of the centers of breeding this perennial grass to make it a viable, economic grain production source as well. And it's tolerant to a wide range of temperature precipitation patterns. So it's well suited to a lot of climates. It actually did, relatively well in the last 2 years here in Minnesota, where we had pretty significant drought. So, so this is Kernza. This is what the breeding has looked like, just to kind of paint a picture for you for how, over the last 40 years, and kind of the progress. So actually the very first variety was only released in 2019. So it's kind of very new on the scene. We've had farmer partners for a long, long time, but only in the last couple of years has someone been able to go and buy seed to plant this. And if anyone's interested after my talk, I also can share with you places online that you can buy flour or the grain to put in to cook with or things like that. So, but this is all like very recent developments, and it's all progressing really, really rapidly also. Yep, it's been popular in bread and beer. It functions a little differently than wheat. It functions a little more like a rye, but it's quite tasty if I say so. And so the other big thing we're thinking about here on top of this ecological, intensification is climate resilience. So, this year we had a big drought, but we are experiencing a lot of flood here. And this is really, typical of climate change everywhere. So Minnesota is experiencing, a wetter, hotter climate where we're in this kind of northern part of the US that's warming faster than other places due to climate change. And so, taking on these challenges is becoming more and more of an issue. And this is just kind of the projection of the two ways that Minnesota's climate is going to go. Either, hot and dry, which is kind of what we've seen the last 2 years. And the year we used to model our treatments was in 1988. And then June of 2014 was one of our very, very wet years, at least during part of the growing season. And so that's how we modeled the precipitation treatments. And another thing we're layering is that however our growing season pans out, we are definitely getting wetter in the winter months. And so we're also simulating this kind of, flood in the springtime to simulate the snow melt, because we're getting more and more snow as we're getting these warmer temperatures actually. So this is the PARSE experiment, perennial and annual responses to climate extremes. This is the overhead shot of this. And so we're rolling out, so instead of Kernza, I apologize, I abbreviated this as IWG, intermediate wheatgrass, which is the name of the plant that the grain Kernza is growing from. And we have 5 blocks in this experiment with these plots and subplots with each of these 4 cropping systems. So we have a monoculture of intermediate wheatgrass. We have an intermediate wheatgrass alfalfa biculture. We have a corn soy rotation, and we have like a friendly, soil health corn soy rotation where we're minimizing tillage and we're cover cropping every year. So those are our 4 systems. And as I said, we rolled those out for 2 years and have just started the climate experiment treatments. So a little more on the climate experiment treatments. The first is this reduction of about 42% of the annual precipitation. And you can see those laid out across the experiment. And then this is what our roofs look like. A point about this is that you'll notice more roofs in the picture than I have drawn on the overhead. And that's because we have 2 different controls in this experiment. And one is an infrastructure control. So you can see in the background the plots that are just these cropping systems growing. And then you can also see some roofs that look more flat in the background, and those are infrastructure controls. And so we have our drought, our flood, our infrastructure control, and then our kind of proper control. And then our other treatment is the flooding treatment. And so those are also being rolled out. And again, you can kind of see that in the middle of that picture in the front, where the drought plots also have an overhead sprinkler, because we're simulating this spring flood before we install the roofs for the growing season. But we can't take them back down again, because of the way that we have the irrigation kind of laid out through the field. All right, and we're again collecting a lot of different data sets, similar to we did in that soil health survey. And so we have all of our plant growth, and in this case, all of our agronomic variables. our soil physical and chemical properties, microbial community properties, where we're looking at both biomass and also the fungal community composition with ITS sequencing, and then the weather or the climate change imposed on this. And we're looking at kind of all of these different combinations, including how the microbial community might be related to changes in the soil physical and chemical properties. And this is our timeline. I noticed a little typo on this in the beginning where it should say 2022, the annuals, or no, it should say 2023, that the annuals are in corn again next year. And so in 2020, yeah, we had successful establishment, but we are not seeing, other than a little change in soil carbon, I'll show you, we're actually not seeing a huge change in the communities in year one. And that's not, horribly unexpected, I would say. It's just the first year. And while we had successful establishment, we were a little bit on the weedy side of our intermediate wheatgrass, which also is pretty common in the first year. It looked beautiful in the second year. So that's been lovely. And then in the second year, the second year before treatments, which was last year, we also had successful growing season. We didn't see a lot of changes in our microbial data sets. Some of our microbial data from then is still backlogged because of issues both with our sequencing center and also some supply chain issues to get equipment fixed in my own lab have also still been an issue since COVID. So we're a little delayed in some of our data sets, but in the second year, we already are starting to see changes in the infiltration rate, the soil water profile, and we're seeing we're not seeing the increase in big aggregates like we did in that other survey, but we're seeing, like, statistically anyways, fewer of the small sized aggregates, which is a trend toward the bigger ones and that development of soil stable aggregation. And we're still seeing changes in soil carbon also. So in that second year, at least we're seeing some developments that we'll be able to follow through in the 2 subsequent years. So these are our carbon results. And yeah, it was really interesting is that in the first year, first you'll notice is that it's actually a decrease in carbon in intermediate wheatgrass, which is kind of surprising to us. And there are some theories about this that I'll share. But yeah, in the first year, we see the significant decrease in carbon in our deeper soil layer in the 15 to 30 centimeters that goes away in year 2. But in year 2, we see a decrease in the 0 to 15 centimeter layer. And we are still kind of puzzling over this, but what I think is happening is and we're puzzling over why the shift from year to year, although you can also kind of see that it's the same trend in year to year, is that in the kind of carbon cycling literature, there's a conversation about carbon storage and how there's this balance with perennial grasses that have these fine fibrous root systems like intermediate wheatgrass does, where it's a balance between how the huge amount of carbon it is putting into its roots versus how much is it respiring carbon out of those roots and how much is it priming the microbial community. And so that could be, that balance as a new system is getting established could be why we're seeing these early changes in soil carbon. Another thing that we've done in this experiment is that even though we're in a pretty rich loamy soil that's great for agriculture, we chose a site that at least had a shallower layer to a gravel bedrock so that in this short duration of this experiment, we could hopefully see, some more changes that are also realistic because of the soil type in general, but so that we could be a little more sensitive to change. So those are our carbon results. And I don't have a whole lot of the results yet because this week even we're taking down the roofs and we're starting to look at things. But one thing from an agronomic perspective, we're trying to weave together improved ecosystem services and climate resiliency with this practicality of something that's economically viable. And so one thing we already were starting to see in late July at harvest was that the kind of harvest timing and impact of that might really be changing. with our climate treatment. So, and you can visibly see how the flooding treatment is visibly still more green than the gold color of the drought treatment. And also, we were in the middle of a drought at this time. And so you'll also notice that our drought treatment looks the same as the ambient, the piece of the crop that's growing outside those roofs. And that's just because of the drought that we had this year. And when we actually were pulling the grain out of here, we had data that matched this color where the grain, in this plots was a lot softer and not ready to harvest yet. So those are just some first inklings of change. And we have taken our soil samples, and then in a couple of weeks, once the roofs come down, we'll be taking deep samples for the root biomass. So that's a look at this big climate experiment. So again, changes have been slow to develop, but they are developing. And then the climate treatments on the impacts of soil are forthcoming because we're kind of just, in the depths of playing out this experiment. So then the third piece I wanted to share is this idea of tracers to better understand carbon flow in different cropping systems. And so we could take these big survey approaches, and they're really, really useful. But at a certain point too, like I was questioning, is this, like, why do we have changes in carbon, and why do we not have changes at other times? And how is this plant functioning? Is it just blowing all the carbon? Is it even priming carbon, and blowing it all off as respiration in this really active root system? Like, what is that looking like? And how does that relate back to a total carbon balance where, storing carbon is kind of one of the things we want to see, both to build organic matter and also to kind of put a drop in the bucket of reducing atmospheric CO2. And so that's where, we're also using more mechanistic studies. And so let's see if my little animation worked. So we labeled this with 13 CO2, and we then traced it into all the above ground and below ground components of these systems, including the soil microbes. And we don't have, and also the bulk soil. So we have our rhizosphere soil, our bulk soil. And this is a study that actually happened in 2020, was all the labeling. And this is a manuscript. If anyone's interested, I can keep you in touch, because this is a paper we're hoping to get submitted in another month or so. We're just wrapping it up. And then this is the chamber that was devised, and this, I'll just take one. I think I'm doing okay on time. I just want to talk about amazing creativity that can come forth, especially from graduate students. So I had inherited this big box, and she completely redevised it. And those coils are her own air conditioner, basically. So these labeling chambers, if it's big enough to kind of encompass the type of system we're working in, can build up humidity and cause a lot of other problems. And so, she basically made a swamp cooler air conditioner to keep the humidity cool. And then she also, you can kind of see on the top the different instruments. She also built her own CO2 meters and temperature meters with a Raspberry Pi interface, so that she could really carefully monitor these systems. And in real time, we could put on the chamber and then watch the CO2 go down to a certain level, so that then we added our tracer back up to ambient. So we didn't have to go above atmospheric CO2 concentrations in order to label. So, and I know I'm only speaking to that and it's not in writing, but if anyone's interested in the methodology and how we devised this thing, I'm happy to tell you more later. And so with that, I'll just dive into some of the results. So this is the milligrams of 13C per meter squared. So the first way of looking at this is just the allocation to above ground and below ground carbon pools. And I think, and so There are a couple of interesting things here. And I'll give you a little bit more about the legend. So IWG1 is 1-year-old intermediate wheatgrass. Again, the intermediate wheatgrass being this grass that produces the kernza grain. Intermediate wheatgrass 2 is a 2-year-old stand. So we wanted to know how is it changing as it ages. And then we compared this to annual wheat. And the green bars are the above ground total biomass, and the brown bars are the below ground total biomass. So, and what we're seeing is, as expected, although more so in the first year field, that we're recovering, we are recovering this carbon, it's being incorporated in the plant almost immediately. And so we're seeing this big pulses and this big recovery, more so in the 1-year-old intermediate wheatgrass and the annual wheat. Like they're really sucking up the carbon right away and also putting it into the roots. But that was telling us that, carbon is being cycled through this plant very, very rapidly. The allocation to above ground biomass was initially higher than that of below ground biomass, but it declined over time. So those are the first results. And then the other interesting thing we're seeing is that the below ground 13C carbon, in the first-year-old field increased, pretty steadily. But we actually saw, when we saw, kind of a not necessarily increase, but a steady, continuation of 13C showing up in the below ground pool of the wheat. But what was interesting in the 2-year-old intermediate wheatgrass field was that this below ground signal decreased really rapidly. And so it's like picking up carbon and then moving it to somewhere else pretty rapidly over this 90-day period. Yep. And just to emphasize that a little bit more, at 90 days, the 1-year-old field still had more 13C below ground than above ground. The 2-year-old field had been kept at above ground and not below ground, although we still picked up something. And then in the wheat, the wheat had been harvested at this point, but we're still seeing that 13C below ground. Yeah, and then our big question is, is it the difference, or what's driving different things in the 2-year-old field? So here is another way that we're looking at this data, the proportion of recovered carbon. And so, we know how much total 13C we were putting in, and we were in real time watching the CO2 concentration. And so we know really, really precisely how much carbon should be going into this system. And so we've calculated the percent of recovered carbon. And the nice, and we could see that, overall we're recovering most of the carbon in the systems. And so, but of course that's changing over time. And so we're recovering almost 90% of the carbon in the annual wheat and the first-year-old wheatgrass above ground right away. And as I was showing in that previous field, it's going down over time. And we see less overall in the 2-year-old intermediate wheatgrass that holds a little more steady through the season. And if we look below ground, again, similar to what I said before, that we're getting this recovery that kind of increases in the 1-year-old intermediate wheatgrass and the wheat, and a lower amount, but more steady amount in the 2-year-old intermediate wheatgrass. We did not, we saw kind of an initial, something's been interesting to puzzle through is that we saw this initial kind of really high recovery of the 13C in the bulk soil. So we saw that in the bulk soil right away, but then it went down in all 3 of our cropping systems, and there's still some, but it's not to the degree of other pools. And then similarly, we're seeing actually a lag where, as like we're seeing less in the bulk soil, and we see this peak in the rhizosphere soil. Well, you can also tell that the rhizosphere soil is really variable, especially in that wheat field. And so where we're seeing more going into the rhizosphere soil, we're not seeing in either of the age stands of intermediate wheatgrass, we are not seeing the same level of recovery of our signal. And then if we, look again, I don't have the legend here, but it's the same colors of the wheat in blue, let me check this one more time. Oh, sorry, the wheat in red, the intermediate wheatgrass, 1-year-old stand in green, and the 2-year-old in blue. Got it. And it's the 2-year-old field where at least we know where it's putting some of its carbon, because this is a 13C lipid analysis, and we're seeing more of that carbon, more of that label in the 2-year-old wheatgrass in these both the total biomass and as well as the fungal and bacterial communities. And so that buildup of the soil community and that kind of development of the biological part of the community are showing up really strongly in that 2-year-old intermediate wheatgrass, less so for the 1 year in the annual wheat. So the conclusions here is that the carbon cycling is really different from a 1-year-old to a 2-year-old stand. There's been some other literature on looking at, really long-term switchgrass fields or other perennials to understand, which agricultural system is going to give us the most carbon drawdown. But we're seeing, at least physiologically, this 1 to 2 years could be a big jump. And in year 2 onward, the microbial carbon uptake might become more and more of a dominant process or an important process in these systems. So those are my 3 chapters. And I just want to give a little shout out to, I've been talking kind of back and forth about economics and the germplasm and all of these things. And so I just wanted to mention a project that is a little newer on the scene. University of Minnesota, as the lead with many, many other partners, was awarded in 2020 USDA CAP grant, which is a coordinated agricultural project through their SAS program, Sustainable Agricultural Systems. And in this grant, we're really weaving together all of these things. And we actually have an objective of integration to make sure that everybody kind of keeps working, to raise this thing together. We have over 81 collaborators representing 11 institutions and 41 other entities. So these are our institutional partners. Just to show their logos across the country. And again, the importance of the community. And so there are these kind of 3 big clusters or communities where there are local brewers or local bread makers or, different other people that are kind of, bringing this thing together. And this picture on the, of General Mills is also a really interesting part of the story, because we are, think, at least on the economic side and with a new co-op around this Kernza grain, they're really trying to develop, a model that can both accommodate kind of local economies and local businesses who might want to buy 20 pounds or 50 pounds of grain. And also, to scale something, we need big players like General Mills. So this picture was taken, at General Mills headquarters, and which is also here in the Twin Cities. And how do we kind of bring those partners along too, so that we can really, change landscapes and scale things. These are my many, many, many thanks. Many collaborators, many students who contributed to the, the chapters I told you, and and a lot of funding sources that have gone into this also. So I'm gonna stop sharing so I can at least see everyone for just 1 minute. And yeah, and now I'm happy to take questions.
Awesome. Thank you, Jessica. We already have a question from Kirsten.
Hi, Kirsten.
Hey, Jess, how's it going?
It's all right.
So fun to hear your research, and I guess at least for me, this is a new line of your research. So, yeah, really fun to see this. Okay, one is just like a benchmark is Iowa State. There was a UC farm where we did the topographic gradient. So that research might be interesting for you to see. We saw similar like erosion collection on the toe slope. Did a bunch of microbial stuff there. So yeah, just thought I'd point that out as like a sister study that might be interesting to compare across.
Yeah, that would be really interesting. Do you, I've not been able to get this chat to function well, but if you don't mind.
I'll follow up online.
If you don't mind emailing me a link, that would be fantastic. Because a piece of that project I didn't mention is that we're doing a really large review of the literature also.
Oh, great. Yeah. So I'll pass you some papers on that, or at least like some keywords so you can find them.
Oh, perfect.
But then I wanted to talk about your studies and the isotope tracer, obviously. So yeah, if I had a question about, I wanted to kind of talk through your above and below ground results a little bit more.
Yeah.
Can you pull that slide up? Because I would love to think through that a little bit more, and especially thinking about new carbon inputs in a perennial compared to an annual system. So the wheat's annual, right?
Yep.
Right.
It's annual spring wheat.
So I'm wondering, and I'm sure you have a long-term plan for this, but I would imagine that so I was thinking about like, yeah, these ones. So I'm imagining that the new rhizodeposition may actually be deeper in the soil profile in the perennial plants, right? So if we think about that new input, I'm wondering where all the new growth is each year, right? And so because perennial plants aren't necessarily making all of the new roots in the same way that an annual plant is, there could be some spatial variability in the depth profile between the perennial and the annual, right? And if we think about that root architecture too, it might be influencing why we see big differences in the first year versus the second year IWG in the below ground biomass, right? So we see differences in the biomass, and then I would imagine that also relates to rhizodeposition and where that might be happening.
Yeah. It's an interesting thought. We weren't, this was such a model thing that we only sampled inside this pretty relatively tiny area. And so we weren't able to, trying to think of how deep she went. She didn't go really, really deep, unfortunately. But yeah, I wonder if it's starting to pump its carbon deeper in the 2-year-old.
Well, because it's really curious
that showing up. An interesting thing too with intermediate wheatgrass is that it'd be really interesting to look at those depth profiles over time, as you're saying, because it, on one hand, it's putting its roots so deep. On the other hand, like no matter how old of a stand it is, like still like 65 to 70% of its roots are still just in the top like 20 centimeters. So yeah, so like, is it still keeping them where the bulk of its roots are, or is it starting to like funnel that carbon down? Would be really interesting to look at.
And then another angle on that same question is like, is there a difference in phenology because the perennial plant already has its architecture in place? Like maybe its allocation is happening. It's like dump of carbon is happening earlier in the season than the wheatgrass. I mean, I don't know, but those were things that occurred to me because you do have pretty striking differences in the rhizosphere soil.
Yeah, huge. And yeah, and that's something that is really possible. Yeah. And I didn't dive as much into the discussion parts of this, but yeah, there's and I'm also not a plant physiologist, which is why I didn't, but yeah, there's it's like an inquisitive versus conservative plant physiological stages. And that's exactly one of the things we're kind of rumbling over and writing about, is that in the first year, it's more of this kind of inquisitive seeking, gathering resources, right? Building the architecture, just like you're saying. And then even into the second year, it's shifting to this more conservative structure where it's, it's still growing, but it's also maintaining and like working toward more long-term growth.
Yeah,
exactly.
And so in this graph too, I was wondering, what is this percent of the 13C delivered? Like you're saying 90% of the 13 CO2 is showing up in that above ground biomass?
It's crazy, isn't it? Yeah.
Well, so then how does that add up with like 70% showing up below ground?
Oh, actually that's a good, now this is making me question how she did that calculation. Right. Because if there's 75% below ground,
yeah, it would be fun to do a budget because it looks like you've got enough of the bulk to do that. I'd be curious to see how that allocation pans out. Yeah.
Yeah, she could. Let me see if I'm gonna stop sharing because I can't look at my speaker notes the way that we did this. So let me see if she has a note about it, and I can follow up with you though because she has done full budgets.
Yeah, I see Vanessa waiting too, so I have other questions, but I can follow up with you.
Well, no, I'm really riffing on the same thing. I'm riffing on the same thing a little bit. First off, those error bars are massive because it's isotopes and soil. So there's entirely possible that things landed funky that way. But I actually wanted to follow up on Kirsten's because that was actually the same thing I was thinking with those deeper roots coming in year after year. Have you done any aggregate analyses on those deeper soils to see if that carbon-13, if there's soil structure forming with the deeper rooted system that might create physical pockets for carbon to be protected?
Yeah, and a little more direct way of answering Kirsten's question. And I'm kind of really, I don't know how many more questions there are, but I'm kind of quick digging through our manuscript here. We were not able to sample super, super deeply in this experiment because when we try to sample more deeply for roots, we go in with a probe truck or other things that are a little more disruptive. And so we, let's see. If I don't find this quickly, I can
That's fine. Don't worry about it because we can follow up later. We know where to find you.
Yeah, and like I said, the manuscript is really close to being finished, and I apologize for not being. I know, especially with the two of you here, I should have been on point with how we did the budgets, but
Jess, this is just
we did not go really deep in the soil in this case. This was more of a first labeling study where we wanted to just make sure that we were careful with what we were able to accomplish.
I'm just really interested in that connection between Kirsten was talking about the exudates getting deeper and then the roots and the exudates combining to create, deeper aggregate structures or physical structure that I think we tend to lose in some of the annual systems. Anyway, I'll leave it.
Yeah, I think so too. And like I said, it's going to be really interesting also because we also, another thing that I didn't show is that in our full budgets, we did look at like what was coming out. And so we had sodium hydroxide traps and we're able to measure the 13C of the carbon coming out. And we definitely saw that increasing over time too. So it'd be really interesting to do that study across the depths and exactly where is it putting it, or even to sample, with like syringes or something to sample the CO2 being released at depth, which is obviously not going to, be very long lived because it'll go anaerobic really quickly. But the fact that it's sending roots deep, but most of the microbial life and like 60% to even 70 or sometimes 80% of its roots are staying at the surface. And so where do plants invest that carbon as they grow physiologically is indeed going to be a really interesting question. And I don't know where I would put my money on that actually, because it seems to be really growing this. Yeah, it seems to be really growing this microbial community too. And so in that case, would you stay up where more organic matter? Would you keep pumping your carbon shallow where there's more organic matter? And you kind of hope and pray that something gets deep where it's going to be stored long term. But yeah, I wonder what it's going to do.